It's a little bit better. Hi, everyone. Welcome to today's live stream. The live stream today is going to be about content category 1C on the MCAT in the Hi, my name is Aiden. I am an MCAT tutor here in New York City who scored a 527 on the MCAT exam when I took it last year. I offer free 30-minute consultations to potential students for which you could sign up on aidenmcat.com. So my first name and then MCAT, MCAT, which stands for Medical College Admission Test.com. The live stream I'm doing is going to be about content category 1D in the Association of American Medical Colleges content outline for the MCAT. Content category 1D is entitled Principles of Bioenergetics and Fuel Molecule Metabolism. This content category pertains to the third section of the MCAT exam, which is the biochem section, so biology and biochemistry. This topic is crucial to not only many topics in chemistry, but also to biology as it explains how our bodies work on the most fundamental level. So I'm going to read the topics contained in content category 1D, and I'm going to share my screen that has the outline for today's lesson, which is taken straight from the AAMC's content outline. And it also has the pictures and the notes that I'm going to add as I explain these topics to you today. Right now, I'm going to read through all the topics so that we have an idea of what we are going to learn today. Content category 1D, the part that I'm covering today, has 1. bioenergetics/thermodynamics. 2, free energy/equilibrium constant, or KEQ, which contains equilibrium constant and relationship of the equilibrium constant and change in Gibbs free energy, or delta for change, and G prime for Gibbs free energy. 3 is concentration, which contains Le Chatelier's principle. 4 contains 4 is endothermic and exothermic reactions. So reactions that either gain heat in terms of the reactants or release heat in terms of the reactants. Free energy, which is equals G, so they're defining G as free energy. The G comes from the fact that it's called Gibbs free energy, after the person who discovered it. 6 is spontaneous reactions and change in Gibbs free energy. 7 is phosphoryl group transfers and ATP, or adenosine triphosphate, which contains ATP hydrolysis, for which the change in Gibbs free energy is much less than 0. ATP group transfers, so transfers of ATP groups from one to another. And then the final topic that I'm going to cover today is the biological oxidation reduction. So this includes half reactions, soluble electron carriers, and flavoproteins. Now what I'm going to do is I'm going to go through each of these 8 topics and their subcategories, and I'm going to explain them, write my notes here, and show pictures that give all of the information that I am explaining. I am going to rely on a series of sources throughout this live stream, so that as much of the information I'm explaining can come from the more reputable and well articulated source as possible. And I'll also add on my way of thinking about it as a 527 scorer and tutor. So the very first topic is thermodynamics. And for this one, I'm going to search up thermodynamics in one of the free textbooks that the AAMC links to MCAT test takers. I think I have a pretty good understanding of it, but I want to first read their definition. Okay, here we go. The definition from the free chemistry textbook that the AAMC likes to use is that thermochemistry deals with the changes in heat during chemical reactions. The primary goal of thermochemistry is to determine the quantity of heat exchanged between a system and its surroundings. Now I'm going to riff off of this definition and include what I think is important to know for the MCAT and for this topic, especially in biochemistry, which this is really a biochemistry lesson. So we're going to start with the first sentence. So thermochemistry is heat or energy chemistry. It is about how much energy this reaction releases or absorbs when it takes place. So it's the heat, and I think of it as the energy of chemistry. There's another domain of chemistry called kinetics, which deals with the speed of reactions, which when you're thinking about a biological system and particularly survival, time becomes very important. Thermochemistry, though, is not the same as kinetics. Thermochemistry is about the heat or the energy of chemistry. So it's heat or energy chemistry. Okay. So the heat exchange between a system and its surroundings. When I think of this, I think of the system as primary reactants and products, and surrounding is everything else. So, in order to determine the energetic favorability of a reaction, which ultimately determines in nature whether or not the reaction is going to occur, we need to figure out what amount of energy is transferred between the system or the reaction and its surroundings, which is everything else in the universe. And when I say reaction, I mean chemical reaction, whereby molecules collide together and form products, new molecules on the other side. This is another way of putting it. Thermochemistry helps us determine whether or not a reaction will occur. Kinetics helps us determine how quickly a reaction will occur. Both thermochemistry and kinetics are going to figure into today's discussion, but this is primarily going to be about thermochemistry. Earlier on, when I was reading all of these bullet points, I mentioned this thing called free energy or Gibbs free energy quite often. That's because it is the way that we quantify energy with respect to thermochemistry. So, heat or energy that we've been discussing this whole time, well, that is Gibbs free energy. That is the number that we determine energy from. It's how we describe energy. So now I'm going to find a good definition of Gibbs free energy. So this here is the definition of Gibbs free energy. Now, the definition demands more definitions because what I have here is an equation that has Gibbs free energy, which is symbolized by G on one side, and then I have these three other variables on the other side of this equation. It's important to start here because these three variables really define what Gibbs free energy is. Gibbs free energy, as explained by this textbook that we're using this information from, is just the quantification of the changes in those other three things. So let's talk about the changes in those other three things. So there is delta H. Delta is that triangle symbol here. Let me get a delta symbol. I'm now going to explain this equation. This equation defines delta G, or the change in Gibbs free energy. The equation reads delta G equals delta H minus T times delta S. Translated, that means change in Gibbs free energy equals change in enthalpy minus temperature times change in entropy. So Gibbs free energy is defined by enthalpy, entropy, and temperature. The only things that can change the Gibbs free energy of a reaction are these three things. Let's start with enthalpy. Enthalpy is otherwise known as the heat of a reaction. Let's find a good definition for enthalpy from our textbook. Okay, here's a great definition for enthalpy. So enthalpy I'm going to change this. So this definition says enthalpy has something to do with the energetic content of a system or molecule. I'm going to change this to enthalpy represents the energetic content of a system or a molecule. Right off the bat, this might seem duplicitous because we've already discussed this value, delta G, which, as stated above, describes the energy of a reaction. And it does. But it turns out that the energy of a reaction is not as simple as just its heat or its energy, the energy content of its system. There needs to be a consideration for this other thing called entropy. Entropy, as defined by this textbook, is how energy is stored. So entropy, they say, has something to do with how energy is stored. I'm going to Google another definition of entropy. Here's the Merriam-Webster definition, part of the Merriam-Webster definition that I want to emphasize in this lesson because it's important to understanding how to solve the problems based on this equation. Entropy is a measure of the system's disorder. The system's order is otherwise known as how the system stores its energy. So does the system store its energy in a way that is very ordered or in a way that is less ordered? That is what entropy is telling us. The change in entropy describes how disorganized that system is becoming from where it starts. Is it becoming more organized or is it becoming less organized? Delta S is telling us the answer to that question. And now we have temperature. Temperature is a measure of the average kinetic energy in the molecules of a system. Temperature in this equation is measured in Kelvin. Why don't we start with a familiar temperature measurement in order to understand what Kelvin is? So Kelvin equals degrees Celsius plus 273.15. For the purposes of the MCAT, let's call it 273. Kelvin is not explained as degrees Kelvin. Kelvin is just Kelvin. So when you're reporting a temperature in Kelvin, it's important that you do not use any units and you just say Kelvin. So if it's 0 degrees Kelvin, that means that it is negative 273.15 degrees Celsius, really cold, the coldest, in fact. And you report the Kelvin value, you say 0 Kelvin as a 0 Kelvin, you do not use the degree symbol. That is the number that we plop in front of the change in entropy in order to find delta G. All right, so there is delta G. It's the energy of the reaction minus the temperature in Kelvin times the disorder of the reaction. And it's the change in all of those things. This is the fundamental, I'd rather say the central, this is the central value that we use in thermodynamics. Delta G, or the change in the energy of the reaction, has a relationship to the concentrations of the reactants in the reaction. So the at equilibrium. So let me articulate that one more time. Delta G has a relationship to the equilibrium concentration of reactants and products in a chemical reaction. So let me find a diagram that might help explain that. Beautiful. Okay. So this is a less accessible diagram, but it has the important part that explains K. So I'm writing now K, otherwise known as K equilibrium, equals products of concentration of the products divided by the products of concentration of reactants, all raised to their stoichiometric coefficients. Okay, let's unpack that definition. I just produced this. This doesn't come from one of the textbooks. So equilibrium constant is equal to the concentration of the, okay, let's wait. Let us, all right, let's fix this a little bit. So that definition was using a more advanced concept called activity, which is more accurate but not relevant for the MCAT. Let's see. That's what all the A's were from. Here we go. Here's an example. So let's learn through example. The equilibrium constant is represented by K or KEQ. That value is equal to the products. So what happens when you multiply the concentration of the reaction products, so these things on the right side, when you multiply those, that value divided by the multiplication product of the reactants, their concentrations, so O2 times SO2, the concentrations of both of those, all raised to their stoichiometric coefficients. So when looking at a chemical reaction, you can find the stoichiometric coefficients by looking right before the molecule or atom. Here it is 2 SO2, 1 O2 equals 2 SO3. If we were doing a balancing equations problem, these stoichiometric coefficients wouldn't be there yet, and we'd have to figure them out. But this this reaction is properly balanced. Yeah. So two sulfurs on this side, two sulfurs on this side, six oxygens on this side, six oxygens on this side. It's balanced. And you balance it using stoichiometric coefficients. So it turns out that this value called KEQ, or Kc for concentration, or just K, is equal to the products divided by the reactants, all raised to their stoichiometric coefficients and all multiplied by each other for the respective sides of the chemical reaction. That value has a relationship to this value, change in Gibbs free energy. And figuring out one or the other helps us get to the other mathematically. So here's the equation that combines those two. This time we're going to go straight to Google Images so that we don't mess up and get any nasty equations from that free textbook, because its figures are pretty awful. Okay, here is the equation. Oh, so let's put that in its proper category. The relationship of the equilibrium constant and delta G. This is the equation for the relationship of the equilibrium constant K and delta G. Look at that. There's one familiar value, that's T, or temperature, and another one which is K, which you just described and defined as this over this. So this is a thermodynamic variable, Gibbs free energy, the change in Gibbs free energy, which, as stated before, tells us whether or not a reaction is going to occur spontaneously, which might as well be, for our purposes, whether or not the reaction is going to occur. We're now learning that that value is equal to negative R, which is a constant that if you were taking the MCAT and given a question like this would be provided to you. So R is just a number. Don't worry about R. Times T, the temperature, times ln, which is the natural logarithm of K. The important thing to take away from an equation like this is not how to plug in numbers and get answers. We will save that for chemistry classes that you took in undergrad. The way to use this kind of equation for the MCAT is to learn the relationships. The relationships. Here, the relationship between one side and the other is that the higher that value, K, the lower the other value. The lower delta G. So once again, higher K equals lower delta G. What does that mean? That means that the more products you have in an equilibrium state, the more favorable that reaction is, because the lower the change in Gibbs free energy is. Why don't I explain a little bit what that means, the change in Gibbs free energy being lower? I'm going to use a diagram. Here we go. So as I said before, delta G determines whether a reaction is spontaneous or not spontaneous. When delta G is less than 0, as it is in ATP dephosphorylation, or hydrolysis rather, as it is in ATP hydrolysis, when delta G is less than 0, a reaction is spontaneous. Another fancy word for that is exergonic. So energy leaving, exergonic. When the change in Gibbs free energy is positive, meaning that reaction goes from less Gibbs free energy to more Gibbs free energy, the reaction is not spontaneous. So it is endergonic. Endergonic reactions can still occur as long as there is an overall decrease in Gibbs free energy of the combined system. So the reactants and what's happening outside. And you can get that by having two reactions go on at the same time, one that's like this and one that's like this, but you got to make sure that the one that's like this is more favorable than this one is unfavorable. So overall, the delta G has to be negative or else it's just not going to happen. No enzymes can change that, by the way. Enzymes are the domain of kinetics, which is about how quickly a reaction will occur. It does not change anything about this that we're looking at here. This is thermodynamics. So it turns out that the more products are favored at equilibrium, the lower the delta G is, and the more favorable is a reaction. Go figure. Because as we just said, a change in delta G determines how the reaction is going to proceed thermodynamically. Is it going to happen spontaneously or is it not going to happen spontaneously? And it turns out it's a let's say like dose dependent relationship. So the less delta G in the lower that value of delta G, so the steeper the decrease in delta G, the more products are going to be favored at equilibrium, and vice versa. The less steep that decrease in delta G, the less the products are going to be favored at equilibrium. Since K is a measure of the products at equilibrium, increasing products here increases the value of K, then we can say that a higher K, more products at equilibrium, equals a lower delta G, and let's say a more negative delta G. That is the relationship of the equilibrium constant in delta G. Note that I did not discuss R, did not discuss T, and did not discuss Ln, because this is about the relationship of that equilibrium constant K and delta G. That is the relationship. Increase K, decrease delta G, more favorable reaction. Next, the concentration of reactants. So as we just said, K is determined by the concentration of reactions. Well, so what's this thing Le Chatelier's principle? Well, it turns out that every chemical reaction is in equilibrium, and you can determine what point in that equilibrium you're looking at by considering how many products you've added versus how many reactants you've added at any given point. You can determine what the reaction is about to do next. So let's get a good definition for Le Chatelier's principle, because it's hard for me, as you can see, to put in words. Okay, beautiful. Here we go. So this equation plus this definition tells us what Le Chatelier's principle is. It's that if a dynamic equilibrium is disturbed by changing conditions, the position of equilibrium shifts to counteract the change to reestablish an equilibrium. So if you took oxygen, otherwise known as O2, out of the system, the reaction would shift to reestablish an equilibrium. If you added O2 to the system, the reaction would shift to reestablish an equilibrium. So all this talk about reestablish an equilibrium, what does it mean in practice? What does it mean in the context of an MCAT question? Well, reestablishing an equilibrium depends upon the reaction favoring all of a sudden one side or the other side. That's how you get back to the point at which there is no change in the concentration of reactants, otherwise known as equilibrium. So, um, I'm going to write now what I think the question is when it comes to Le Chatelier's principle. So, in this example, adding oxygen is going to shift the reaction to the right, because if you put more than the equilibrium value of oxygen onto this side, that oxygen needs to now be converted to this side in order to get all the values back to equilibrium. If you add SO3 to this side, it's going to go left, which means that you're going to be turning some of that SO3 back into oxygen and SO2 in order to reestablish equilibrium. This is the core of Le Chatelier's principles questions. What direction is the reaction going to reestablish equilibrium? The next point that the AAMC asks about is endothermic and exothermic reactions. Let's get a good definition for that. I think it's pretty self-explanatory, but endothermic. Straight out of that textbook, here's the definition. Endothermic and exothermic. So let's remember what endothermic and exothermic mean from our discussion of enthalpy. So it seems like these terms, endothermic and exothermic, refer to enthalpy, whereas endergonic and exergonic, which I introduced earlier up here, and exergonic, endergonic, are referring to Gibbs free energy. So rather than Gibbs free energy, we're dealing with enthalpy here. So exothermic reactions are favored. Exothermic reactions are reactions in which enthalpy decreases. So enthalpy being delta. I should relearn that keyboard shortcut. Delta H is less than 0 equals exothermic. Delta H is, we're just going to copy this, and then it's going to be the opposite. So delta H is greater than 0 is endothermic. So I'm going to now copy this delta G equation again and explain why we can have these two definitions at the same time. So we talked earlier about how favorability is determined by delta G. Well, what's this whole talk about delta H and favorability now? Exothermic and endothermic and favorability. Well, it turns out that since a more negative delta G equals a more energetically favorable reaction, you can say that a more negative delta H is going to equal a more thermodynamically favorable reaction. Negative delta H, negative delta G. If delta S is positive. So as long as things are getting more spread out, which is generally how the universe trends, the reaction and the reaction is negative in terms of its change in heat, you're going to get a spontaneous reaction. And spontaneous reactions are what we need for our body to make the products that it needs to be. It's what it is. 5, free energy equals G. Yeah, there we go. Free energy equals G. That's the definition of free energy. 6, spontaneous reactions in delta G. As I explain up here, delta G being positive or negative determines whether or not a reaction is spontaneous. In the body, that is the same. Our body is just a big bunch of molecules where sometimes those molecules collide together and react. Well, are they going to make the right products, or are they not? And the right products determined by nature are the ones that result from energetically favorable reactions, aka exergonic reactions. And it doesn't have to be just one exergonic reaction. It could be an endergonic reaction and an exergonic reaction happening at the same time, as long as the exergonic reaction is greater in its decrease in Gibbs free energy than the endergonic reaction is in its increase of Gibbs free energy. So you got to have an overall negative Gibbs free energy, or else it's not going to happen. Phosphoryl group transfers and ATP. So let's take a look at what they mean by phosphoryl groups. There we go. This is a phosphate group. There's phosphorus in the middle, and then 1, 2, 3, 4 atoms on the outside. It has an overall -3 formal charge because each of these oxygens has more than its typical number of valence electrons, and it is a group that is passed between biological molecules all the time in our body. The most famous one of these biological molecules is this guy. I'm getting a diagram of it. And here we go. Adenosine triphosphate. Adenosine triphosphate. ATP. So this is the energy currency of the body, as it so happens. This is a nucleoside triphosphate. So here is ribose. Here is adenine. And together these are called adenosine. And then this is a group of phosphate groups. So these guys strung together, triphosphate. 1, 2, 3, 3, triphosphate. Adenosine triphosphate. Adenosine here, triphosphate here. That's the energy currency of the body. But what does that mean? That means that ATP hydrolysis has a really negative delta. I think it's this one actually. There we go. Delta G. That's the keyboard shortcut. Okay. ATP hydrolysis has a really negative delta G, and that drives otherwise unfavorable reactions in our body. So our body is using the sun's energy to basically push reactions in an uphill battle against their natural decrease in energy. And in order to do that, we have to have something else that's decreasing in energy, and that is ATP. All the time, we're taking ATP, we're taking it from the more unstable, i.e., energetic form, ATP, and we're turning it into the more stable, i.e., unenergetic form, ADP, or adenosine diphosphate. So we're making triphosphate into diphosphate. How do we do that? We use water to split the bond between the second and the third phosphate. And that a water splitting reaction in chemistry is called hydrolysis. So ATP hydrolysis has a really negative delta G that drives otherwise unfavorable reactions in our body forward. So in order to do all of that fun, unfavorable chemistry that we want to do in our body, we need to make a sacrifice energetically. And that sacrifice that is being made constantly, all the time, every day, every night, is ATP hydrolysis. We need it in order to survive. The next point of, let's make sure we covered this topic well. Phosphoryl group transfers in ATP. Yeah, so phosphate groups, they go from ATP and they go elsewhere. You can take a phosphoryl group from ATP and you can plop it on to another organic molecule such as glucose. and you can keep it in the cell that way to make it ready for glycolysis. And that's one of the steps in glycolysis rather. Let me grab a picture of glycolysis to show. Oh, wonderful. Okay, Khan Academy it's the best visuals ever. Okay, so here in Khan Academy, we see that the very first step of glycolysis requires that we take ATP and turn it into ADP, thus turning glucose into glucose 6-phosphate, which keeps it inside of the cell. The reason you don't see glucose 6-phosphate here is because they're emphasizing the bigger steps in glycolysis, and they're not showing that intermediate. But let me find a picture of it. Yeah, it's not so pretty. I mean, that's glucose, right? It's a hexose, right? 1, 2, 3, 4, 5, 6 hexose, um, aldohexose at that, and then you add the phosphate group. So there's the phosphate group. It's no mystery where that comes from. It comes from the end of ATP. So glucose to glucose 6-phosphate. Let's get the free energy change of that reaction. There's another picture of the same reaction. So we go from glucose, we add ATP, hexokinase, which is an enzyme that speeds up this reaction that we're not going to talk about because we're not covering glycolysis today, is in the middle there. That's an enzyme, aka a protein that speeds up chemical reactions. All right. So here's a sentence from Google. The direct phosphorylation of glucose is energetically unfavorable, requiring an input of roughly 14 kilojoules per mole. So the kilojoules per mole is the unit that we use for enthalpy and also Gibbs free energy. Pretty sure. Let's just make sure about that. Standard units. Yeah, so for both delta G and delta H, the unit is kilojoules per mole. And then for delta S, it's a different unit, but that gets converted into kilojoules per mole in this equation so that we can properly subtract T delta S from delta H to get delta G. So the product delta G is in kilojoules per mole. Well, it turns out that plus 14 kilojoules per mole is the energy change going straight from glucose to glucose 6-phosphate. That can't happen spontaneously because that is a positive Gibbs free energy change, and positive Gibbs free energy changes are not spontaneous. So how does this reaction happen? Well, check this out. Under standard biochemical conditions, ATP hydrolysis releases roughly 30 kilojoules per mole or 31 kilojoules per mole. So about 31 kilojoules per mole. Well, 31 is a bigger number than 14, which means that the reaction, when both of these reactions are happening together, the hydrolysis of ATP and the addition of a phosphate group to glucose, that means that the whole reaction overall is spontaneous, because overall you have a decrease in Gibbs free energy. You add these together and you get a negative number, roughly negative 16, and boom, the reaction happens spontaneously. So ATP hydrolysis, indeed, yes, AAMC, that delta G is less than 0. It's not the most energetically favorable thing ever, but it is energetically favorable enough to make that the energy currency of our cells. So that Gibbs free energy change powers our cells' reactions. ATP group transfers. I want to just make sure what that means. I think I know what it means, but let's double check. Oh, okay. So yeah, an ATP group transfer. Okay, so the substrate of this reaction is glucose. So phosphorylating the substrate in this way using the phosphate group from ATP is an ATP group transfer. I don't know why they call it ATP group, because we're not transferring whole ATPs. We're transferring the P, one of the Ps, to make it ADP. Okay. The final topic is oxidation and reduction. Whenever oxidation and reduction comes up on the MCAT, I repeat the mnemonic to myself that oxidation is loss and reduction is gain. Taking the first letters of those 3-word statements, oil rig. Oxidation is loss, reduction is gain. When it comes to oxidation reduction, we talk of half reactions. Let's get a good diagram for a half reaction. I can explain it with words, but it's better seen. So first I'm going to write oil rig. And then we have an example of an oxidation reaction. So let's see the overall reaction here. So the overall reaction, this chromium molecule with oxygen plus iron 2 goes to chromium plus iron. So you're taking chromium with a bunch of oxygens and you're turning it into chromium. And in the process, we're removing an electron from iron, i.e., we're oxidizing iron. So iron is getting more oxidized, chromium is either getting oxidized or reduced. Let's see. So the half reaction is defined as the reaction where either oxidation or reduction is occurring within the greater reaction. So every oxidation reduction reaction is both an oxidation and a reduction, because when you have two molecules and there's going to be some oxidation reduction happening, one of them is going to be the oxidizer, one of them is going to be the reducer. So in any such reaction, you can split out the oxidizer and the reducer. So here the oxidation half reaction, iron is going to be oxidized. It's 6 Fe 2 plus goes to 6 Fe3 plus 6 electrons. So from every Fe, you're taking 1 electron and you're turning it into Fe3 plus. The loss of electron is oxidation. This CrO7 compound, Cr2O7 2 minus goes to 2 Cr3 plus. In this case, we're not losing any electrons. We are gaining. Are we losing any electrons or gaining electrons? Well, let's see again. So overall, it is CrO7 2 minus. So what we have over here. Oh, well, it's a reduction reaction. So it has to be the gain of electrons by definition. So here, CrO7, 2 minus. I guess I was just confused about which species is gaining and which isn't. Here, it's chromium is getting its electrons back. They used to be consumed by that oxygen. So the reduction is gain of electrons. Chromium is going to gain its electrons. Now, iron is going to lose them. So the part of that reaction that's oxidation is oxidation half reaction. The part of the reaction that's reduction is a reduction half reaction. Altogether, we have 6 iron 2 plus chromium 2 chromium, 7 oxygen, 2 minus, plus 14 protons, which is otherwise denoted as H plus 6 electrons, goes to 2 Cr3 plus 7 H2O plus 6 Fe3 minus, 3 plus rather, plus 6 electrons. So taking the electrons on both sides, they balance out. And that is always the case in a chemical reaction. You never have a change in the total number of electrons. The electrons just don't just go flying into space. They stay in the reaction. Okay, so that is half. These are the half reactions in general. But let's take another look at these molecules that I'm talking about here. These are not organic molecules. Okay, these are not the molecules that we're talking about in biochemistry. These are what I'm using to illustrate the concept of oxidation reduction, which goes throughout all of chemistry, not just biochemistry. So let's get a biochemical reaction here to explain this concept. Okay, so here we're not dealing with half reactions because we have just the reduction and oxidation of one molecule. We're not splitting them out. So NAD plus is, let me triple check the full name of that molecule. It is nicotinamide adenine dinucleotide. So nicotinamide is this molecule that adds on to an adenine. And let's see. We have a picture here of the structure of NAD rather. Nicotinamide adenine dinucleotide here. Okay. So yeah, there's adenine. It's the same adenine we see over here, part of ATP. There's adenine. Here is nicotinamide, and here is another ribose sugar that is making this a dinucleotide. So we have the nitrogenous base. This time it's nicotinamide. It's not one of the ones that we see in DNA or RNA, but it's a nitrogenous base. So this is a dinucleotide, nucleotide because it is sugar, phosphate, nitrogenous base. Sugar, phosphate, nitrogenous base. Two of them, dinucleotide, nucleotide. So nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide and its partner, FAD, which is also an adenine dinucleotide, are, this is the part of FAD that we're talking about here, are the electron carriers of our body. They're two of the most important ones. They're also coenzymes. So they need to work with an enzyme in order for an enzyme to catalyze the chemical reaction that it is catalyzing. But what does it look like when we take this idea of redox and apply it to one of these very important molecules in the body? Well, here we have a hydride. The hydride is the hydrogen, so it's thus the proton, the isotope of hydrogen that is seen most often in nature. We're not talking about deuterium. We're talking about one proton. So it's a proton and no neutron, which usually you would have an equal number of neutrons in every other element. But for hydrogen, it's usually just one proton in nature. So we got the proton, we have 2 electrons. So it's a hydride. It's a negative formal charge because rather than balancing out the positive charge of the proton, there are 2 electrons bringing with them 2 negative formal charges. Though that hydride attacks, using organic chemistry terms, it attacks the benzene ring on nicotinamide in nicotinamide adenine dinucleotide, and in NAD, and that becomes NADH. So the H adds on, the H gives its electrons, and now this is the reduced form. Reduction is gain of electrons. So NAD to NADH. is a reduction reaction. The opposite reaction also occurs all the time. It is an oxidation reaction. The carrying of electrons via NAD, NADH, and FAD and FADH2 is the process through which we get our electrons through the electron transport chain. At the end of the electron transport chain, we donate those electrons to oxygen, which is the final electron acceptor. That's an important fact to know for the MCAT. And that is the process by which we start to produce a lot of ATP. Let me get a good diagram there. So the final electron acceptor in oxidative phosphorylation is indeed molecular oxygen, otherwise known as O2. What else comes along with that molecular oxygen? H2O. Or two hydrogens, so it's H2O. And that wouldn't be molecular oxygen. It would be one oxygen atom for every two hydrogen atoms. That makes H2O or water. So these are just the numbered complexes of the electron transport chain. At the end, the electrons plop down onto somewhere. Usually you would have 2 electrons at a time, which would mean that you have a full O2, accepting those electrons, 2 protons, and then you get water as the side product. So mitochondrial water is responsible for some of the water content of our body. So our body produces its own water in small part. So the other important thing that happens here is that as you're passing electrons down the electron transport chain, you pump protons out into this side of the mitochondrial membrane, and those protons are going to fall down back into the inside of that membrane via this guy, which is the pump that at the end of the electron transport chain turns ADP and phosphate into ATP. So oxidative phosphorylation ends in the phosphorylation of ADP to make ATP, and that is done through this complex. ATP synthase. Yeah, ATP synthase. Just wanted to double check that one. Okay. All right, so these are the soluble electron carriers. We have NAD, NADP, and FAD. So here is a screenshot of the Google results for that Google search. NAD, NADP, and FAD. When I didn't mention NADP before, it's just NAD plus a phosphate group. And then flavoproteins. Well, flavo, the F in flavo comes from flavin. So let's find a picture of. Okay, so riboflavin, otherwise known as vitamin B2, is a vitamin, the derivative of which can add onto a protein to create a flavoprotein. So, what is the importance of flavoproteins in this discussion? Well, let's find a couple of examples. FAD is one of them, as is succinate dehydrogenase, which is a complex in the Krebs cycle, glucose oxidase, cytochrome P450 reductase, cytochromes, cryptochromes, rather, and phototropins. So they throughout the body, this derivative of riboflavin is used to make all different kinds of things. One of them is this coenzyme FAD or FAD2. The flavin comes from the flavoprotein part comes from flavin adenine dinucleotide. So that covers the first part of. So we covered all of these topics. That covers the first part of content category 1D, the principles of bioenergetics and fuel molecule metabolism. So what about when we take all of these concepts? I'm just giving a heads up now to the next parts of content category 1D. What happens when we take all of those concepts of delta G, delta H, delta S, temperature, the concentrations of reactions and products, K, the equilibrium constant, endergonic and exergonic reactions, And then these electron carriers, phosphate groups, ATP, NAD, NADP, and FAD, H2, and FAD, and apply it to the reactions that actually do occur in the body. Well, here's one of them. We stumbled upon one of them while we were discussing this. That's glycolysis. So the MCAT demands that you remember the intermediates and the enzymes that act in these reactions. They're not all particularly high yield, but some of them are, based on which ones are regulated by the body and are important steps in regulating the possibility of these reactions happening in the body at any given point, because that needs to change depending on the body's circumstances. So there are a few of these that are necessary to know for the MCAT, and that will be a different discussion, but that's just a heads up. So applying all of this that we learned about energetics, thermodynamics, we learn how our body makes the molecules that it needs, how it takes glucose, which is that first energy molecule that comes from our food, and turns it into the energy that we actually use in our cells to live. There are a bunch of other processes, but they all use these similar principles of a negative delta G and transfer of electrons using electron carriers. Thank you for watching today's live stream. I hope it was helpful, and good luck as you prepare for the MCAT.